Good afternoon, everybody. I think we'll get started. For those of you who don't know me, my name is Mike Verneris. I'm in pediatric BMT, and I'm one of the co-leaders of the THI program. It's my pleasure today to introduce Mike Leibowitz, who is a new faculty member here at the University of Colorado. Mike did his undergraduate work at Bowdoin in Maine, and then went to medical school at University of Pittsburgh, where he did an MD and a PhD. And then he came here to the University of Colorado, the Colorado Children's Hospital, for pediatric residency, internship and residency training, and then back to the East Coast to the University of Pennsylvania, this time Children's Hospital of Pennsylvania, where he's been doing really interesting work looking at chimeric antigen receptor T cells and targeting the stroma as well as the tumor itself, which is what he's going to talk about today. I'll ask you guys to be kind to Mike because he spent the whole day so far giving talks. He gave a talk in our BMT session earlier today, and now he's giving a totally different talk here. So Mike's been with us for 6 months, and he's been an absolute pleasure and a great addition to our program. And so he'll be applying to the Cancer Center as a mentored member. He has funding from Alex's Lemonade Stand and St. Baldrick's, which are two pediatric focused, probably the two largest pediatric focused funding agencies, and is going to be writing a K coming up soon. So without further ado, Mike, take it away. You might need to turn this on.
All right. All right. Thanks, Mike, for the kind introduction. Before I get started, I just want to make a shameless plug. If you guys know anyone that's interested in joining a lab, or you yourself want to join a lab, be open to having people come. It's been a challenge finding folks. So if you know anyone, please send me an email, let me know. So today I'm going to be talking about utilizing CAR T-cell therapy to treat solid tumors. And the strategy that we're going to take is increasing epitope spreading. The majority of the work that I'm going to be talking about today was performed at my prior institutions. Let's see if I can, there we go. So I know we're a bit of a mixed audience, and I just wanted to make sure we're all on the same page in terms of thinking about CAR T cells versus conventional T cells. I think we all are aware that conventional T cells recognize antigen in the context of MHC, which is a major limitation in terms of adoptive cell transfer, whereas chimeric antigen receptors can recognize cell surface antigens without the context of MHC and built in. Let me see if I have a pointer. You guys see that? Does that pointer show up? Okay. And within that SCFV, that antigen binding domain, there's co-stimulatory molecules, so you get both signal 1 and signal 2. But again, one of the major advantages of CAR T cells is you can recognize antigen without requiring MHC. It's non-MHC restricted. Just wanted to start off with a little bit of clinical background. So many of you guys may know or know of Emily Whitehead. She was the first pediatric patient to be successfully treated with CD19-directed CAR T-cell therapy. She was treated at Children's Hospital of Philadelphia. She had multiply relapsed refractory B-cell leukemia. She received CD19-directed CARs, became quite ill, was hypotensive, had fevers into the 104s, 105s. She developed what we now call is CRS, or cytokine release syndrome. Steve Grupp and Dave Tichy threw everything at her, etanercept, anakinra, and so forth. She didn't get better until they gave her tocilizumab, which is an anti-IL-6 antibody. She quickly defervesced, was off pressors, and is now probably about 10 years cancer-free. Really, the success in using CAR T cells in Emily and in the adult side at Penn really led to a revolution in using cellular-based therapies to treat relapsed refractory diseases. And currently, there are multiple FDA-approved indications of using CAR T cells in hematologic malignancies, but we haven't been able to translate that same level of success in solid tumors. And the question is why? We all have our favorite cartoons. This is my cartoon that I drew. It's certainly not inclusive. There are many barriers to successful CAR T cell therapy in solid tumors, but these are some of the major ones that I wanted to emphasize here today. So one is trafficking. So unlike in hematologic malignancies, where CAR T cells can encounter tumor cells in the blood, in the lymph nodes, in the bone, oftentimes solid tumors can be in immune privileged sites or sites where it's difficult for immune cells to actually translocate to. There can also be physical barriers. So a lot of solid tumors recruit cancer-associated fibroblasts or CAFs, which can secrete stroma and also secrete immunosuppressive factors like IL-10, adenosine, IDO, which can down-modulate immune responses. So if the CAR T cell is able to circumvent those barriers and actually translocate into the TME, it has to deal with a very harsh microenvironment. So typically the microenvironment is low pH, nutrient poor. There are immunosuppressive cells like regulatory T cells that down-modulate immune responses, and hypoxia is becoming a bigger and bigger issue. There's a lot of metabolism studies in looking at cancer therapy now. But the barrier that I'm really going to focus on today is tumor antigen heterogeneity. I think it's a barrier that's understudied and definitely requires more attention. As a side note, I think it's very important that CAR T cells proliferate, persist, and avoid exhaustion. And those are also sub areas in the CAR T cell therapy field that a lot of people are investigating. And I also think it's really important that CAR T cells induce a broad endogenous anti-tumor immune response. I don't think CAR T cells as a monotherapy will be successful in solid tumors, and I'll tell you why. I really like this picture. I show it all the time. I think it's nice. My kids think it's nice. They do listen to my talk. That's how I put them to sleep. So this is a immunofluorescence of a lung adenocarcinoma. And as you can see, some of the tumor cells stained with mesothelin in green, some with CA125 in red, and you can see many of the tumor cells don't express either antigen. So you can imagine if you have a CAR T cell that targets one or both of these antigens, you're going to be left with a significant amount of tumor, and you're not going to get a complete response. So I think this really highlights tumor antigen heterogeneity, and I think it's also important to note that CAR T cells only target surface antigens. So that led us really to ask a couple key central clinical questions. Can CAR T cells eradicate tumors that don't uniformly express the CAR T target antigen through induction of an immunologic bystander effect, which surprisingly is an area that has not been investigated all that much? And can we alter that TME to enhance CAR T cell therapy for solid tumors? So this is a cartoon that Carl June and Michael Satterline drew, I guess now, 3-4 years ago, that I think is more of a hypothetical. We don't really know that this occurs. So when we infuse our CAR T cells, we presume that our CAR T cells do in fact kill a proportion of the tumor cells. Those tumor cells then die, release neoantigens, and if they're functional antigen presenting cells within that TME, in theory, maybe they'll cross-present those antigens and activate endogenous anti-tumor immunity. So this is a process called epitope spreading, and I think an important immunologic bystander effect that we would like to have happen in solid tumor CAR T cell therapy. So that led us to try and test as to whether this immunologic bystander effect occurs. So I think we developed an appropriate model, and this work has been published. I don't think I put the, well, anyway, it's been published, where we took a mouse mesothelioma cell line called AE17. We modified it to express human mesothelin, so we called it AE17 meso. In collaboration with Novartis, we developed a high affinity, highly potent CAR that recognizes human mesothelin. This is a second generation CAR with a 4-1BB, and we cloned this into a retrovirus. And as you can see in the flow, we got pretty good transduction efficiencies, and it was highly potent at killing human mesothelin expressing tumor cell targets in vitro. So we wanted to ask the question, what percentage of tumors need to express the CAR T target antigen in order to eradicate tumors? And can CAR T cells induce an immunologic bystander effect? And the one that we're most interested in looking at is epitope spreading. So we did these mixing studies where we took the wild type tumor cell line without human mesothelin, and we mixed it at defined ratios with the CAR T target expressing tumor cell lines. You'd think this would be very easy, but actually developing this model is a little bit challenging. And the reasons are you need to have a CAR T cell that's quite potent and can eliminate tumors where 100% of the tumor cells express the CAR T target antigen. And it was very important to perform this in immune competent mice. A lot of the work that was done to develop the CD19 CARs are done in NSG, and you're really losing large components of the immune system. And we're, of course, interested in looking at immunologic bystander effects. And I think the model should avoid radiation and chemotherapy. It can affect immune cells adversely. There are probably people here or on the Zoom call that use radiation to actually promote inflammation, and it may have a positive benefit, but at the time when we were thinking about this, we didn't want to use any radiation or chemotherapy. So what we did in our first study is we injected tumor cells subcutaneously into mice where 100% of the tumor cells expressed human mesothelin. We gave 2 doses, 10 to the 6 transduced CAR T cells 2 days apart. And as you can see, we were able to completely eradicate the tumor. However, when we injected mice with tumor cells that did not express the CAR T target antigen, you can see the tumors grew, and ultimately these mice died. Okay, so what we did next was we developed tumors where only 50% of the tumor cells expressed the CAR T target antigen. As you can see, you had some tumor growth inhibition, but ultimately these tumors grew out and the mice died. When just 25% of the tumor was composed of tumor cells that lacked expression of the CAR T target antigen, again, we got some inhibition, but ultimately these tumors grew out. The mice died. And even when just 10% of the tumor was composed of tumor cells that lacked expression of human mesothelin, you can see we got pretty good growth inhibition, but by about day 19, these tumors began to grow and ultimately the mice succumbed. So what that told us was, at least in this particular model, with this particular antigen, with a 2nd generation CAR and a 4-1BB, you can see there are a lot of caveats, that CAR T cells were not able to eliminate tumors in which not all of the tumor cells express the CAR T target antigen. And if this is true and holds true in other models, it would suggest that CAR T cells as monotherapy probably wouldn't be effective in treating solid tumors, in part due to antigen heterogeneity. So how can we overcome this issue? So that led us to hypothesize, and please, anyone ask questions in the middle, I don't mind, and if something doesn't make sense or isn't clear. But that led us to hypothesize that in order for CAR T cells to be effective in solid tumors, we would need to alter that tumor microenvironment and or induce immunologic bystander effects. So that led us to investigate 2 strategies to do this. One is modulating the TME through the use of immunostimulatory chemotherapy. We use a lot of chemotherapy as oncologists, and actually there's quite a bit of literature showing that chemotherapy at different doses can affect immune responses. So we're very interested in that. And then the second strategy was to enrich for dendritic cells. So there's a specialized subset of dendritic cells that are very good at cross-presentation, and I'll get into that. And our hope was to increase epitope spreading as an immunologic bystander effect to make CAR T cells more effective in our solid tumor models. All right, so the first chemotherapy we're going to talk about is cyclophosphamide or Cytoxan. It's a chemo that's been around for a while. We actually use it as part of our lymphodepleting strategy pre-CAR T in the clinic. But interestingly, there's a whole body of literature showing that different amounts, doses of cyclophosphamide can actually have immunomodulatory properties in terms of affecting dendritic cells, depleting regulatory T cells. So we wanted to see what effect combining this chemotherapy with our CAR T cell models could potentially have in eradicating tumors. So just to remind everyone, even just 10% of the tumors that lacked expression of our CAR T target antigen ultimately grew out and the mice died. So we wanted to test whether given Cytoxan pre-CAR T cells could improve this phenotype. So what we did here was we took mice, we injected them with tumors where just 10% lacked expression of the CAR T target antigen. We injected our CAR, we gave 100 per kilo IP of cyclophosphamide, or we gave Cytoxan in 2 doses of our CAR T cells. And in blue here, you can see that we were able to eradicate the tumor. So that was pretty cool. And we wanted to sort of figure out the mechanism. What is happening? Why is this occurring now? So an easy experiment that a lot of folks do is you take knockout mice of various immune components and you see, does the phenotype go away? And not surprisingly, in the absence of CTLs, we lose that phenotype. So CD8s are important, not terribly earth-shattering. I had mentioned that Cytoxan depletes Tregs, or at least it's been reported in the literature. When we looked in the tumor-draining lymph nodes, so these are tumors in the flanks, so you can look at the inguinal nodes, which are the draining nodes. So when we extracted those and we looked for regulatory T cells, we saw a significant decrease in regulatory T cells in mice that received cyclophosphamide. Systemically, it didn't have a significant effect, but you could see systemically in the spleen, we did deplete some Treg cells, and within the tumor, we were modifying the microenvironment, and we did see a significant decrease in regulatory T cells. I mentioned epitope spreading a bit. In our tumor models, we're able to modify them to express chicken ovalbumin, xenoantigen. I know a lot of people poo-poo that, but I think it's a nice model in that we have tetramers that have been validated that work quite well. So when we looked at our mice, so each column represents a different mouse. Each row represents the different treatments. If you pay attention to the bottom row here, you can see we had a significant increase in ova-specific T cells in our mice treated with both Cytoxan and our CAR, suggesting an increase in epitope spreading. Moving on, in the interest of time, I wanted to talk about gemcitabine. This is a pretty cool chemo. We don't use it all that much in pediatrics. I think it's used a lot more on the adult side to treat non-small cell lung cancer, pancreas cancer. Occasionally, we use it a bit in sarcomas in the pediatric world, but like cyclophosphamide, gemcitabine has also been shown to have multiple effects on the immune system. There's data to suggest that it decreases Treg activation, depletes MDSCs, and can actually skew tumor-associated macrophages more towards an M1 versus tumor-permissive M2 phenotype. And there are some data that gemcitabine can actually increase T cell infiltration and DC cross-presentation in tumors in various publications that I've listed here. So that got us a little bit interested in playing with gemcitabine a little bit. So like cyclophosphamide, we decided we wanted to alter that tumor microenvironment before giving our CAR T cells. So on day 6, we gave a single dose of gemcitabine IP. We gave 10 to the 6 transduced CAR T cells. This is a different model. This is a mouse lung cancer model where not all of the tumor cells, once again, express the CAR T target antigen. Although we weren't able to cure the mice, and some of these studies I want to continue here, even though it's lung cancer model, but we're able to inhibit tumor growth quite nicely. And if you look at day 25, you can see we're able to significantly inhibit tumor growth. But perhaps more importantly, we had increased survival in these studies. So moving on, I want to talk a bit more about enriching dendritic cells as a way of inducing epitope spreading in our solid tumor models. And I want to talk a little bit about dendritic cells. They're pretty cool cells. They kind of, like everything in immunology, come in vogue and then kind of go away and come in vogue. To me, they're in vogue. But basically, these are professional antigen presenting cells. In order for them to fully mature and activate T cells, in order for them to fully activate T cells, they need to be mature. And maturation markers include expression of CD80, 86, CD40, class 2. And interestingly, dendritic cells, when they interact with regulatory T cells within the tumor microenvironment, can actually cause DCs to be energizing. So it's actually a bad thing to have Tregs and DCs together within tumors. So how can we augment epitope spreading? And again, I think it's probably through this theoretical thing that I had mentioned before, cross-presentation. So dendritic cells, like a lot of immune cells, are complex. There are many different subsets. Turns out there's a subset of dendritic cells called conventional dendritic cell type 1, or CDC1s, that are quite good at cross-presentation. The issue with CDC1s is they are very rare within tumors. So a lot of folks have looked for these specialized dendritic cells in a number of different tumor sites and tumor types, and they're quite rare. Although the presence of these CDC1s within tumors do correlate with improved outcomes in a lot of different tumors. So that got us thinking about, well, how can we enrich for these dendritic cells within the tumor microenvironment? So turns out there's a growth factor called FLT3 ligand. For the oncologists in the room, that's usually a bad thing to have that in the setting of AML. But when you're trying to enrich for dendritic cells, it's a good thing. So FLT3 ligand, it's secreted by stromal cells, to some extent NK cells and NKT cells. The receptor for FLT3 is actually expressed by both lymphoid and myeloid lineages. However, it's lost in the lymphoid lineage and just expressed by dendritic cells during development. And FLT3 ligand has been shown to be very important in DC homeostasis. So a number of folks now, it's been 5-6 years, have made FLT3 ligand and exogenously injected them into mice. And you can see, based on these CyTOF studies, they got enrichment of these specialized cross-presenting dendritic cells. They're also called CD103 positive DCs. Like immunology, there's 10 different ways of identifying the same cell, but you can see it worked quite nicely here. About 3-4 years ago, Stephanie Spranger and Tom Gajewski's lab published a very interesting paper showing that these CDC1s actually secrete CXCL9 and CXCL10, and were able to show that they were able to enhance recruitment of both adoptively transferred and endogenous T cells within the TME. So these dendritic cells not only would participate in cross-presentation, they would recruit immune cells as well. So that got us pretty excited, and it led us to ask the question, well, what would happen if we armed CAR T cells to secrete FLT3 ligand in terms of altering that tumor microenvironment? And could we enhance anti-tumor immunity in our models? So what we did here was we made a construct in which our M11 CAR would in fact secrete FLT3 ligand. Again, it's a second generation CAR. Here you can see the transduction efficiency was not changed by virtue of secretion of FLT3 ligand. In vitro, we're able to detect lots of FLT3. The killing between our FLT3 secreting and non-FLT3 secreting CARs were equivocal, and activation and secretion of pro-inflammatory cytokines like interferon gamma were similar. So they seem to be functioning similarly to our non-FLT3 CARs in vitro. So that led us to ask the question, well, do they actually enrich for CDC1s within the tumor microenvironment? So we injected our mice with tumor. We gave 1 dose of CAR, either our FLT3 secreting CAR or non-FLT3 secreting CAR. And about 3 days later, we resected the tumors, digested them, and wanted to identify CDC1s. So this is the gating strategy, and it's actually pretty complex, and you need to get a lot of cells because, like I said, these dendritic cells are very rare. So we get it on live cells, we selected leukocytes, we excluded macrophages, T cells, B cells, NK cells. and then ultimately gated on 11C positive. And we had two markers to distinguish CDC1s versus CDC2s. So XCR1 denotes CDC1s, CD172 alpha CDC2s. I'm not really going to get into CDC2s. It's another subset of dendritic cells that aren't quite as good at cross-presentation, so we're a little less interested, but nevertheless, we pulled those out as well. All right, so when we looked at the tumors, what we found was a twofold increase in CDC1. So that was pretty exciting compared to our non-FLT3 secreting CARs. So we wanted to ask the question, well, do our CARs get into the tumor? Are they functional? Are these DCs doing anything? So we did a pretty cool assay where we resected the tumors. These are live tumors, not fixed. We put them in agarose, and then we made thin slices of these tumors. We stuck them in vitro in complete media just overnight, and we assayed for different cytokines and chemokines. On the x-axis, these are different thickness sections. And as you can see, in terms of FLT3 ligand, we saw more FLT3 ligand in mice that received the FLT3 ligand CAR. So that's great. They seem to be trafficking to the tumor and they're secreting FLT3 ligand. We also assayed for CXCL9. Again, that's a chemokine that these CDC1s secrete to attract endogenous and adoptively transferred T cells. And we looked at different thickness sections. We did see a thickness-dependent increase in CXCL9. So that was cool. Again, we're inferring that the CXCL9 was due to an increase in CDC1s by virtue of increased FLT3 ligand within that tumor microenvironment. So I'm kind of pivoting here. So when I was at my former institution, I was doing mostly lung cancer type studies because my PI was mostly interested in that. As I've said before at some of these talks, you don't see a lot of primary lung cancer in kids. I'm a kid doctor, and we do see a lot of bone cancer in kids. So got interested in osteosarcoma. Just as a little bit of clinical background, it's the most common malignant bone tumor in children and young adults. Kids don't do well, even with localized disease. They die about 30% within 5 years due to metastatic disease. The 5-year OS for patients with metastatic disease at presentation is less than 40%, so it's not good. The standard of care, how we treat these patients, typically is with chemo and surgery. We don't do a lot of radiation. These tumors tend to be radio-resistant, and really the standard of care for osteosarcoma has not changed in 40 years. We've been doing the same stuff. There are also very few salvage options. So if you have a patient that relapses or recurs, there's really not a lot of options or experimental therapies out there. So for all these reasons, I really wanted to focus a bit on osteosarcoma. Also had a patient with really bad pelvic osteosarcoma, and there weren't a lot of options for him. So it got me interested in this type of tumor from a research perspective. And I'll also just mention the COG, which is the Children's Oncology Group, is prioritizing novel agents for osteosarcoma, realizing there are very few options. So it's a good field to get into because there's a lot of black boxes. All right. So again, the models that I like to use are immune competent models as opposed to NSG models. And again, as an immunologist, I like to look at how the immune system is affecting things. The problem with osteosarcoma, there are very few syngeneic osteosarcoma models. We got one from the ATCC called K7M2, developed by, I think, Kamal Khanna like 20, 30 years ago. And this is a metastatic tumor cell line derived from BALB/c mice. It was from spontaneous MET. And he developed a cell line, which we could get through the ATCC. I gave one of these talks at Baylor, and Jason Eustein, who's a sarcoma specialist there, interested in osteo, let me know that he developed two murine osteosarcoma cell lines from black 6 mice with a TP53 mutation, which is commonly seen in human osteosarcoma, and he kindly shared that with me. So we have those as well. We modified these tumor cell lines to express luciferase so we could follow growth and treatment responses via IVUS. So this is an example of a mouse. You can see we injected it into the tibia. So these are orthotopic implantations, and you see metastases. They quote 90, 95%. It's not that high, but nonetheless, they do metastasize to the lung. And we were really interested in treating metastatic disease because that is what causes the worst clinical outcomes in our patients. We're interested in looking at FAP, or fibroblast activation protein. It's a serine protease. I mentioned cancer-associated fibroblasts in the beginning of the talk as a cell that deposits stroma and also secretes immunosuppressive factors. So fibroblasts, activated fibroblasts, cancer-associated fibroblasts express FAP. There's been a lot of data looking at FAP showing that it is associated, FAP expression is associated with tumor progression, metastases, recurrence, and a poor prognosis. It's very common in epithelial cancers as well as desmoplastic cancers. It is expressed during normal embryonic development and typically isn't seen in normal adult tissues. So in fibrotic conditions, we do see FAP expression like IPF or a cirrhotic liver. And in fact, when I was at Penn, we had a collaboration with cardiology looking at cardiac fibrosis, and we injected our FAP CAR T cells, and it got rid of the cardiac fibrosis, which is kind of cool. And in fact, Mike, I was interested in looking at this in pulmonary fibrosis and I was going to talk to you about that. I just remembered that. Anyway, yeah, so what we did here was, so FAP is expressed in cancer-associated fibroblasts, but it's also been shown to be expressed in tumor cells themselves, like osteosarcoma. We took TMAs at CHOP, and we did the IHC looking for FAP, and we did see FAP mostly in the periphery of the tumor. But we also see FAP in other solid tumors, pediatric solid tumors, like Ewing sarcoma, hepatoblastoma, even a little bit neuroblastoma. You see it a lot more in hepatoblastoma, perhaps due to the fibrosis and stroma. Hepatoblastoma is very rare. All pediatric tumors are rare, but hepatoblastoma is very rare, so I didn't go down that route too much. So at Penn in Ellen Pure's lab, they developed a CAR T cell that targets FAP. And this FAP CAR T cell also recognizes mouse, human, and dog. Looking at various tumor cell lines, admittedly, these are lung cancer cell lines. You can see when FAP was expressed in these tumor cells, you got decent growth inhibition, but ultimately these mice died. So it seemed like these CAR T cells were specific. And this was a paper from 2014, so this was a bit ago. But they also did second harmonic generation microscopy to look at fibrillary collagen. And you would expect if the FAP CAR T cells are decreasing stroma, you would see less signal, and that's exactly what they saw here. So it seems like the FAP CAR T cells were specific. They were hitting the stroma, which again is a barrier to CAR T cell therapy in solid tumors, in some solid tumors, I should say. All right, so we wanted to ask the question, well, what happens when we use our FLT3 secreting CARs in our tumor model? And I wouldn't say surprisingly, because nothing, I guess, is all that surprising in science, but we did not see the phenotype we were hoping to see. We were hoping to see, because we were able to increase these DCs, we're able to increase CXCL9 in these tumors, that we would have seen growth inhibition or cure in these tumors in which not all of the tumor cells express the CAR T target antigen. We didn't see that, so that was a little disappointing. And for the graduate students in here, there's going to be a lot of disappointment. You move forward, keep on working, and you'll get through it, hopefully. Like I said, despite enrichment of CDC1s within the TME, we didn't see improved tumor regression from our FLT3 secreting CARs. So why is that the case? It made us really pause and think hard. I mentioned that regulatory T cells can alter dendritic cells and make them energizing. And again, going back to the prior data that I showed you, at least in terms of Cytoxan depleted the regulatory T cells. So there's a huge regulatory T cell component in this particular tumor model. I didn't have time to show you, we also did the same studies using Dreg mice. For those of you that are familiar with that, it's basically a diphtheria toxin receptor gene upstream of FOXP3, and you could selectively ablate regulatory T cells by giving diphtheria toxin to these mice. And what we found was by virtue of just depleting Tregs in this particular model and just giving our CAR T cells, we're able to induce cures. So this particular model is very Treg dependent. So it made us hypothesize, could it be that these dendritic cells, even though there was a higher percentage, they seem to be secreting some chemokines, could they be defective in terms of antigen presentation? So we went back and we looked at the CDC1s and we phenotyped them. So we looked at a lot of different maturation markers, CD80, 86, class 2, and we found not necessarily that they were down, but they were not up. And it led us to hypothesize that perhaps these dendritic cells were defective in terms of antigen presentation, co-stimulation, and we weren't actually activating T cells. So that led us to look through the literature and try and sort out, well, how can we induce DC activation and maturation? And there's a number of different approaches. So one approach is using poly-IC. I believe there are even clinical trials using poly-IC, and there are a lot of papers out there showing that they can activate and mature dendritic cells. Type 1 interferon, so interferon alpha is another strategy. The one that got me kind of interested, I've been interested in agonistic anti-CD40 for some time. My background, way back when I graduated from college, I worked with Bob Vonderheide at Penn, and at the time, he was very interested in using CD40 to activate B cells and mature B cells as an APC in breast cancer and other models. And agonistic anti-CD40 has been around for a long time. I think Randy Noel might have cloned it first. Don't quote me on that, but it's been around for a long time. There are issues with toxicity. It has lots of pleiotropic effects on the immune system, but there are also very promising clinical studies that are underway using agonistic anti-CD40 in the setting of pancreas cancer at Penn. And given my interest and whatnot, and the fact that we were able to show that CD40 did upregulate these co-stimulatory molecules on our CDC1s, and the fact that it's already in clinical trials, it got me interested in using agonistic anti-CD40 antibody. So what it does is it licenses DCs, they secrete IL-12, and it supports priming of CD4 and CD8 T cells. So the first thing that I did was injected CD40. I injected a lot of CD40 because that first experiment, you want to see a phenotype. So we gave CD40 on day 9, 12, 15, which is quite a bit. And then we sacrificed the mice about 2 days later. We did see already an effect on tumor growth just by virtue of CD40 by itself. When we looked at regulatory T cells, we saw a little bit of a depletion. But interestingly, we did see a lot of OVA-specific T cells, suggesting that some form of epitope spreading was happening, whether it was the dendritic cells, maturation of dendritic cells, or some other APC, we didn't know, but it seemed promising enough to move forward. So what we did here was we took our CAR, and we combined it with CD40, the idea being ultimately use our FLT3 secreting CAR in combination with CD40, giving the CD40 after to mature the dendritic cell. And we wanted to ask the question, could combining CD40 with our CAR increase our therapeutic efficacy? And it did. So that was pretty exciting. We wanted to look at epitope spreading in combination with our CAR, very small percentages, but we did see increases in ovis-specific T cells in the tumor-draining lymph nodes. And I think this is really the definitive study. So what we did here was we did an interferon gamma ELISpot assay. I'm sure most people in the room have done this. And what we did was we took the spleens from mice that were treated with CAR CD40 or CAR and CD40. We isolated the CTLs, and then we co-cultured them with our tumor cells. And basically, every spot represents an activated T cell. And we saw many more spots in our combination. Again, we don't know exactly what tumor antigens these T cells are recognizing, but the simple fact that we were able to increase tumor antigen-specific CTLs were important to me. So here we go. I think that's sort of the definitive study. So next steps, I am kind of shying away from lung cancer for the reasons that I mentioned and moving more towards osteosarcoma. And we plan to do a bunch of preclinical studies in our osteosarcoma mouse models using our FAP-FLT3 CAR. I didn't get time to show our FLT3 FAP CAR and the data behind that. And we were looking at combination studies with agonistic anti-CD40. Very interested in looking at changes within the TME to characterize the tumor immune infiltrate by flow and other mechanisms, and to assess epitope spreading. Of course, we want to look at survival analysis and look at therapeutic efficacy. And I think it's important to sort of look in models that are closer to humans other than mice. So it turns out that dogs spontaneously develop osteosarcoma, and I think also have that TP53 mutation and is very similar to human osteosarcoma. So called up Dan Regan, who I learned about during our I guess summer retreat, fellowship retreat, and he is going to be looking and assessing for FAP expression in these osteosarcoma dog models. And again, the CAR that we have can recognize human, dog, and mice. So I think that's pretty powerful. So kind of putting it all together and what we think and hope will occur is that we have these CAR T cells, they secrete FLT3 ligand, they enrich for these pre-CDC1s, which come from the bone marrow, in combination with CD40 to activate and mature these dendritic cells. So again, the CAR T cell will recognize a proportion of those tumor cells within that TME, kill them, these tumor cells will release antigen, these mature dendritic cells will then take up those antigen, cross-present, secrete these chemokines to recruit both Mike's favorite cell, NK cells. Actually, there are data to show that these CDC1 specifically recruit NK cells, so maybe we can talk about that too. And endogenous T cells. And then hopefully these CDC1s will then prime sort of a broader anti-tumor immune response to eradicate all those tumor cells that are left that the CAR T could not recognize because they don't express the target antigen, leading to tumor regression. And I know this is ambitious, this is what we hope will happen, and we hope to get those studies underway. And again, one more shameless plug, anybody knows anyone that wants to work in this field or thinks this is moderately interesting, please give me a call or email, and I'd love to have you join the lab. And finally, just want to acknowledge a few folks. So John Maris has been a great mentor at Penn. He's more of a neuroblastoma guy, but let me work a little bit on osteosarcoma in his lab space, which is fantastic. Steve Albelda, who's the lung cancer CAR guy at Penn, who I worked with as well. Nick Olimpo, who's now a graduate student here. Maybe some of you guys know him. He worked with me right out of college and really great. I'm sure you guys love him. And he's here. A few other postdocs. Ellen Pure is a fat, straight PhD hardcore biologist and is brilliant. Bob Vonderheid's been a mentor for 20 years and is fantastic. And I didn't really get to show the data from Wayne's lab. So Wayne Hancock is the Treg specialist who we collaborated on the DREG, DTR mice studies. And again, acknowledge funding. And of course, I should amend this and thank Mike and Leah and other folks here also for their support. With that, I am happy to take any questions, and I'm supposed to put this up where you guys can crush me. Yeah. Thanks. I think that's a great question. Certainly in the hematologic realm, we know that the CAR T cells need to persist, because if they don't, we see these B cell precursors called hematogones. In the setting of solid tumors, I don't know the answer to that. If we're able to produce a sufficient endogenous anti-tumor immune response, pardon me, we may not need persistence. My inkling or bias would be persistence would be important, but I don't know the answer to that. It's a good question. Not you. Yes.
Okay, so I'm just reading questions out from the chat.
Oh, sorry, Mike.
Would you also, when you say your questions, will you repeat them so people on the chat can hear them?
Sure. Am I able to see the chat questions or
I
no?
have one for you.
Oh, okay.
Did you try CD440L instead of anti-CD40L?
I have not. No. In part, have I tried CD40 ligand instead of agonistic anti-CD40 in our studies? I have not. I think in part, we had expertise in using agonistic anti-CD40 antibody in Bob's lab and Greg Beatty's lab. So I sort of went with the expertise that we had, but it's certainly something we could look at. And whoever sent that question, send me an email. I'd love to chat more. Should I get Mike here really quickly? All right. Should I get my boss? Sorry.
So, next
Yeah.
question. So,
Yeah.
you might have said it at the beginning and I missed it, but, as you see spreading immunity or epitope spreading, right? But one of the ways to really prove that is either re-challenge the mice, or to take splenocytes to treat established mice. Try to do those things?
Yeah. So, to answer your question, no. But the one data to suggest epitope spreading, or the one paper that I'm aware of, she left Carl's lab. I can't remember her name, but she used the EGFR, I think V3 CAR. I can't remember her name. Apologize. But I believe they were able to cure mice and then re-challenged with the same tumor and saw that the tumor did not form. I think I have mixed thoughts about that particular experiment. I think it's something we should do. I mean, I know folks sometimes put tumors, on the contralateral side and modify one versus the other, but I think you're futzing with the immune system too much. But I think it's definitely a re-challenge study is definitely something that we should do. What was the second way?
Transfer splenocytes.
Or transfer splenocytes. Yeah, definitely could do that. I think that makes a lot of sense. The question is transferring splenocytes by themselves or in conjunction with CARs. But I think that's a great thought and something we could do. Patricia. Okay.
Kind of does it but is it better if it's locally produced than it's served? Like, I don't know how.
Yeah, it's really, really interesting. I think Dave Tuvison's group, like 2-3 years ago, at least in the pancreas model, really looked at location of immune cells within that tumor microenvironment. And I think those are really good questions. I have not injected systemic FLT3 just because it is really expensive. So I think it might be something that works just as well as local, but in my mind, having the CAR in there, having that local FLT3 where you do get induction of these dendritic cells, the CAR is destroying tumor cells, releasing antigen, that physical proximity. I mean, I can't speak to it because, you know, we're digesting tumors, although with these slices, these tumor slices, you can, and I think Greg Beatty's group, I should reach out to him, developed some IHC where you can sort of determine where that lymphocyte is in relation to the dendritic cell and the tumor cell and so forth. So I think proximity and physical location is definitely something worth investigating. I don't know if I answered your question. Yeah, any of the students have questions? No, okay, all right, yes. Yeah.
And sorry, I forgot to remind you.
Oh, sorry, yeah.
So can you comment on the whole pediatric microenvironment, like dog, mouse, human model, dendritic cells? So are dendritic cells in your model are all adult animals, right? So
Yeah. Yeah, all right, so the question was, can I comment on the different microenvironments in dog, human, mouse, pediatric versus adult? I think that's a great question, Patricia. I mean, we're using mice that are 6 to 8 weeks old. We're not using really old mice. We're not using very young mice. I think certainly we're all aware that there are differences in immune systems and immune responses in mice versus dog versus human. I think a lot of us use mice because they're relatively cheap. We have some models, but I honestly am not totally aware of the differences in the immune responses or immune systems in dog versus human versus mouse. In terms of osteosarcoma, I mean, it is seen in adults. It's sort of a bimodal distribution. I think 50-year-olds or so in adults, don't quote me, I'm not a medical oncologist, get osteosarc. And there might even be differences there as well. I think those are all things definitely worth looking into and could sort of enrich more than just a therapeutic study. What are the actual immune differences there? All right. And Nick did a lot of this work. Nick's great. So be Nick's friend. All right. Thanks, guys.